right. Good morning and welcome back, everyone. A little bit weird to be back on campus. I had entered through a different door and I can honestly say I got lost. So hopefully your morning went a little more smoothly. Um, so our schedule's a little bit mixed up now, with missing a day, and then I forgot to include the day of Truth and reconciliation on Thursday in the schedule. But luckily, this chapter should be we can finish up today and Thursday, and then we'll start chapter 5 after that. So we have enough wiggle room that we should be all right. I will say I'm so close to having the midterms graded. I have one question left. I'm get it done this morning, and it just didn't quite work out, and I didn't want to rush it. So I'll do that as soon as I get home after this class. So you should see the mark today. I'm going to be releasing the full exam to you guys. Usually, I prefer to do an in-class or not an in-class, like a face-to-face exam viewing, just to kind of keep those questions to myself. But with online and everything else going on, it's just so much easier to release everything to you guys. So you'll be able to see all your questions and your responses and the correct answers. And then for the written responses, any comments that I've added, you'll see those as well. So hopefully that is a good study tool for you. And then from here on out, we're hoping that they'll be in person as planned. But I've learned not to anticipate too far in advance. I think that was everything I wanted to talk about. So we can launch right into chapter 4. And this is an interesting combination of methods of research and also methods of research. And it's kind of strange. I have a bunch of other classes at differing levels in psychology, and they all do methods and ethics of the second class. So right off the bat, first or second week, that's when you launch into methods and ethics. But in this textbook, they took some time coming around to it, and I'm very glad that they did, because when we start getting into some of the methods, we now have that base understanding of how neurons work and how the brain works. It allows us to make sense of what they're talking about. So I might be adjusting my teaching methods in the future because I kind of really like this layout. But most of this hopefully is going to be fairly repetitive and review-like to what you see in other and probably just other, probably bio as well classes. I don't know how much overlap we'd have with something like chemistry, but we can talk about broad things like theories. So a theory is going to integrate and interpret diverse observations in an attempt to explain a phenomenon, which basically means it's going to take all of the observations, all that we know about a particular topic, and try and explain it using a unifying concept. So a theory is going to be our overall explanation of what we think is happening. And a good theory should lead us to create testable hypotheses. So if we have a theory of how a certain part of our brain works, we might form a hypothesis that says if we deactivate that part of the brain, say using PMS, which we'll talk about later, we should see this kind of behavior. So that would be a testable hypothesis that's based on our theory of how that part of the brain works. So that's pretty straightforward. We should have a basic understanding of theories and hypotheses. The textbook also does one of the best jobs I've ever seen of explaining why scientists speak the way they do in their publications. You might have noticed if you've read scientific papers that scientists default to language. It seems like, it appears as though, it might be such that. And you might wonder, why aren't they just saying that this is what happened? Right? Why don't they like the term proven? Why do we sort of dance around saying caused something else? What's going on? And the author of our book actually does a good job of explaining that it's not that we don't trust the science. It's that over time, the process you've learned that what we think is happening, what we think is going on, through the best of our ability at the moment, might tomorrow have some new evidence shown that. And as scientists, it's our job to think, great, new evidence, we can change it, we can be better. So these theories and these hypotheses that come from the theories evolving and changing. So we acknowledge that there might actually be flaws in our studies. There may be flaws in the methods that we're using. A lot of stuff that we're going to talk about, especially in this class, we might have thought that things worked one particular way until we developed a way to study it. Until we had the tools and the technology to study it, until we figured out we were completely wrong. So there's lots and lots of cases of that, especially with neuroscience. And we get new data that's coming in, so more people are doing studies in different ways, and maybe our theory holds for the data from one study, but when another study comes at that same problem from a slightly different angle, we have to adapt our theory to explain those new results. That's why stuff is always changing, always moving. And even if we do a good study, and even if we do have good techniques and everything goes well, when we're making conclusions, when we use statistics, we use that 95% rule, P of 0.05, where we say that we accept that there's about a 5% chance that everything that we've observed is due to naturally occurring. So there's a possibility that even a good study has a significant result that isn't there. So we keep all of this in mind, and that's why good science is usually tentatively stated. So when you see a lot of newspapers or say Facebook or something like that, say scientists have proven this thing, that's usually a very expanded and over proposed thing. And that's going to tie into our assignment for this course, which I posted the details for, but I'm not going to go over in this class. I'm going to do it in the next class, depending on timing. But if you wanted to sort of glance over that, the assignment's going to involve finding the scientific literature that's been reported in the media and evaluating some of those claims that they're making in the media articles. So we'll talk at length about that a little later on, and I want to include some references to things like APA formatting, because I know that everyone has used that by the time they get to this course. So that was a tangent I did not intend to go into, but that's all right. When we're talking about our methods of study, we have two very broad categories that our research into. We can have experimental studies and we can have correlational studies. Now, correlational studies are actually a subcategory of a broader group called descriptive studies or descriptive research. But for our purposes, we don't really care about any of the other kinds of descriptive research except for correlational studies. So descriptive research might be, say, a headline reports that 50% of students rather stay at home than go to class. That is a done by collecting survey data, and they can report that of that particular population. But it's not actually a correlation because for a correlation you need two measured things that are sort of varying together. So that's a little bit of a technicality there. I teach methods course, so I really like specifying. Correlational is a subpart of descriptional, just so you know. But so we talked earlier on about the fact that science and research in general is all based on empiricism. So we base our conclusions, we base on observation. And for scientists, observation has a massively broad image. We can talk about surveys, like I just mentioned. Maybe you send out emails and ask people to complete a survey online. Maybe you stop people randomly in the hallway and ask them to rate something. We can also look at case studies. So look at an individual or a group of individuals that have experienced a unique situation, and see if you can draw any conclusions or infer any relationships from what they experience. We can have actions where you just observe things in nature. So maybe you sit outside one of the doors on campus and you count how many students go by in a particular time slot. That would be a naturalistic observation. And then, of course, we have experiments, which we really like, but can't always do. So let's start with our experiments because they're one of the easier things to sort of conceptualize. So when something is an experiment, we specify the researcher is manipulating some variable. And the variable that we're manipulating is our independent variable. That's because it's not going to change unless it is independent of everything else that's going on, hopefully. And we're trying to see if that independent variable influences our dependent variable. We want to know, does the dependent variable depending on the level of the independent variable? So we're trying to do manipulations, and you can have multiple independent variables, you can have multiple dependent variables. There's lots of different iterations we can get into, but mostly we look at one-to-one experiments here. And we want to be aware of the fact that things, especially in psychology, very rarely exist on their own. There's usually something else going on. We're looking at behavior. It's not necessarily just somebody's mood. Maybe it also depends on how hot the room is, or how uncomfortable their shoes are, or whatever else you can think of. So we want to try and eliminate those extra variables, those things that could be potentially confounding variables, in order to make sure that we're only looking at independent, dependent relationship between those variables. So we're going to control as many things as possible, and we can do that in a bunch of different ways. We can use it through the design setup. So maybe we want to use a repeated measures study instead of a between group study. So you have everybody, instead of having sort of an experimental group and a control group, you would have only one group of participants, and they get the first treatment and then the second treatment, and you measure all the way through so they can be their own controls. That would be one way where groups begin with group. You've controlled for those extra variables. Or maybe you can't do something like that. It has to be two distinct groups. So maybe you're going to collect a bunch of data, age and sex and whatever else might be relevant. And you can add those variables into your statistics to make sure that they're not varying between your groups. So all of these extra controls, all of these extra steps that we take mean that we get sort of a better type of representation of that relationship between our two variables, but we're sacrificing some of the realism. So by holding all of these things constant, we're looking at a very artificial environment, and that's not necessarily the easiest to apply to the real world. So you can study someone's in an MRI as you like play sounds at them, but what does that tell you about someone walking around in the world hearing things around them? So we have to keep those in mind when we're looking at those results as limitations of studies, especially with brain research. I worked with someone who he got sick and tired of the fact that most brain research was done stationary in enclosed environments where you couldn't do anything. So he's rigged up a setup that you can wear on your back. And he gets people to go bike riding through the river valley and looks at their brain activity while they're cycling. So if that's something you're interested in, that is Kyle, Kyle's last name, Mathewson, Dr. Mathewson at the U of A. So he does some cool stuff there. But that's another tangent, and we'll come back to EEGs in a minute. For our correlation study, so instead of manipulating a variable and seeing how it changes another variable, in correlational studies, we're just going to be measuring two variables. And you can do multiples. Again, we're not to just two, but we look at the simplest case. You can do much larger correlation studies with, say, regression, looking at 5, 6 more variables, but you really don't want to unless you have. So the goal here is to look at two different variables, see how they change over time, see how they vary, hence the name. And then we want to check that variance between those two measures like as they go together. So if we looked at, say, height and weight, a really easy one. So as people tend to be taller, so you measure a bunch of people, measure their height and weight, taller people tend to also be heavier because they are bigger. They are taller, so they have more mass to them. So we could do a correlational study of height and weight and probably find a positive correlation in most populations. That doesn't mean that everyone who is taller is going to be heavier than someone who is lighter, but we're seeing a general trend here. And so we want to keep in mind that because we're measuring these two things sort of in the nature of things, and we're not manipulating them, we're not changing anything, we don't control that setup. We've lost a lot of control, so we can no longer make decisions about cause and effect. If we were doing something where we physically changed a trait, we had someone who was given a particular drug, and we measured how it changed. If that drug is the only thing we changed, we see that the drug caused a change in behavior. But if we are not manipulating that variable, then we can't make that causal determination. We can only say that they're related, and we might infer that one causes the other, but we can never be certain. There's a little bit of a caveat there. If you go deep enough into statistics and do an extensive amount of research, you can tick enough boxes that it can be reasonable to make the assumption that something is causal. This is an aside beyond what you need, but if we look at, say, the claim that smoking causes lung cancer, that is one of those things where people are like, oh, well, that's correlational. You can't experimentally force people to smoke cigarettes and see if they get lung cancer. And that's absolutely right. But they've done enough experiments coming at it from enough different directions that they can check all the boxes that we get from doing an experiment. So they've done correlational studies that look at sort of over time, same people across different time periods, different people across different time periods, and more. They've studied it inside and out, and that's why that's one of those things that I don't necessarily mind seeing it causes something else, even though they haven't done an experiment. But that's one of those with an asterisk, and you have to know what kind of research they've done to trust a correlational study saying that something causes something else. We also want to keep in mind some other limitations here, other than not being able to get those causal relationships. There's a chance for confounding variables. Again, there's other stuff that could be going on. In an experiment, we try and control for those things. In a correlational study, we're just measuring. We can't necessarily control for those things. Again, there are statistical methods where if you measure all of those things that might be confounds, you can pull them out using regression methods, but that's really complex. So mostly we just want to be aware that that could be a problem. And so we take from this that the results from these studies can be really, really helpful, and they can give us a strong indication of relationships between variables, but we have to be really careful about how we interpret that information. So don't assume a causal relationship, and be aware of things like confounds that they may not have taken into account. So if you're reading a study, you might want to watch for what else they've done in their statistics, what else they've pulled out, what else they've accounted for. And then this is just a sort of graphic from our textbook that shows the difference between correlational studies and experimental studies. So at the top, they're saying that they've observed individuals with frontal lobe impairment also tend to have violent criminal behavior. And since we've measured those two variables, we haven't caused damage to that area and observed for aggression. So we're saying that, okay, well, maybe that damage to the brain causes the aggressive behavior, but maybe their aggressive behavior led them to receive damage to their brain. Maybe because they're aggressive, they were injured more often. So there could be sort of either direction involved here. Or more likely in a lot of cases is the possibility of a third variable. So maybe there's something else that causes both frontal lobe damage and violent criminal behavior. So something like drug use or physical abuse can lead to both of those things. So they might just be sort of incidentally related. That third variable could explain the relationship. And that's why it's so hard to say that A causes B, especially when we don't know what else is out there. With an experimental study, it's a lot simpler because we induce a change in our independent variable, control for as much else as we can, and we see an adjustment in our dependent variable, so we have that assumption of causality. Mhm. Sorry, what was the first? Yeah, so and it would still be like you have to be aware of your limitations there, where I think where I've seen quasi experiments are like you can't assign children to be in a particular classroom or to have a particular socioeconomic status or whatever. But as long as you balance your groups okay, you should be able to account for it. And it's always there's always floating asterisks around of you can make causal statements if, and that's sort of if you go on into statistics and methods, there's a whole checklist of things that you need to hit in order to do a statement. So we draw a hard line, and it gets blurrier as you get higher. Yeah. All right. Okay, so I think that's all I wanted to say about our basics. As I said, I've had a bunch of different classes that have very similar topics, so I'm not sure how much of that is overlap with what we've done already and not. But I really enjoy this chapter, so we'll keep on going, and we're going to talk about some of the research techniques that are commonly used when looking at the brain. So this is going to be a lot of overlap with sort of cell biology because of staining and imaging and stuff like that, as well as all of the psychology stuff that we've been learning with parts of the brain and how the brain works together. So a lot of these techniques are going to be how we figure out how the brain works and how it together and how things function together. And again, all of these techniques are constantly changing, constantly improving and advancing. And even since I was doing research, I used to do DNA sequencing in single celled organisms. And in the 10 years that I had not been doing that, the field has changed dramatically. And they no longer have to send supplies out. You can just do it in house because it's a lot more affordable, it's a lot faster, and it's a lot more accurate nowadays, too. Um, all right, so first we're going to talk very, very briefly about how we go about staining and imaging neurons. So if you've thought about it, all of these neurons, all closely packed together in the brain, are all basically the same substance. They're all cells and they're all intertwined. So even though we think of them as sort of here's a neuron and here's another neuron and there's another one, it's a lot more dense and twisted and compacted. And so when researchers finally got microscopes and they were able to look at cell tissue, it was sort of just a confusing grayish mess where they couldn't distinguish where one cell began and another ended. So that's an issue with resolution. So how small can you get into things to see them? But also, is there any contrast between different structures? So if you've done any bio classes involving staining, you know that there are tons of different kinds of staining. There's lots of different ways of labeling particular components so that they stand out from other components. We're only going to be talking about four basic kinds, and we're going to keep it pretty high level because each of these could get its own chapter on processes. Um, the first of these we can talk about is the Golgi stain, and I actually have images for some of these. So this first one here is a Golgi stain, and it's going to highlight individual neurons. So it randomly stains about 5% of the neurons on a slide. The textbook doesn't elaborate on how it does this randomly. I think it has to do with which have a particular gene active at a particular moment, but I didn't have time to look into it, I'm afraid. They just keep it high level of at random, about 5% are highlighted. And that's perfect because we can see that one really, really clear one in the middle, and everything else sort of fades into the background. And that's because all of the other surrounding neurons are not highlighted. And so here we can see our cell body and our dendrites and our axon. You get to see all of those components. And it's a lot messier than maybe some of the drawn diagrams that we get to see. This is what they would look at to determine what the shape is of particular neurons in particular areas. The next of our stains are the myelin stain and Nissl stain, and these guys operate kind of in opposites. So you compare them together and look at opposite parts of the cell. So the myelin stain gets taken up by the fatty myelin tissue that surrounds our axons. So that's going to highlight all of the white matter. And then the Nissl stains is going to bind to the cell body, so the places that aren't myelinated. So that's going to get us our gray matter. So we can look at, again, on the next slide, the top, the myelin stain, but showing all the white matter on the inside here where it's darker. And then on the bottom, we have the missile stain, which is the cell bodies or on the outside. So pretty neat that they're able to highlight in opposite ways. So another way that we can image the brain and take a look at the structures. We also have retrograde stains. And these are really neat because we're finally starting to see just a little bit of movement here. So instead of just looking at an extracted piece of tissue and looking at where those cells are and what the shape is of those cells and sort of highlighting just the outside of those cells, now we're looking at stains that get taken up by the neuron terminals and get ended up transported into the axons, into the cell bodies. So these are going to only be taken up by cells that are active. Make sure I say what I wanted to say. So there's a couple of different options. The textbook talks about fluoro gold as the most common one. I don't really care about the specifics on that particular one. But this allows us to use sort of the fluorescent qualities of any of those stains that get taken up into the cells that are active to tell us which part of the brain receives signals from other places. So it tells us which ones are active. So if we trigger a particular pathway, we can see the cells in that pathway pulling up that stain and activating. So that's when we start inching towards getting an understanding of function. And again, as promised, going into tons of detail on these, and that's perfectly fine. All right, so the next two that we want to talk about are autoradiography and immunohistochemistry. And I'm surprised that went as well as it did considering the time of day. But as I said, those earlier staining procedures and the tracing procedures along that pathway are great for showing the fine anatomy, don't really give us a ton of detail about function. So we're not getting a ton of moving parts, as it were. So in autoradiography, we're going to be staining those cells as well as revealing which ones are active. So it's kind of a dual purpose thing. So we stain just like the other ones, get that structural component, but we also have activation when those neurons are actively firing. And so the textbook describes a couple of different ways to do this. I'm actually going to talk sort of anecdotally about one that our lab uses fairly often. You don't have to know the specifics of this, it's just to illustrate how it works. We use a particular setup, a long, boring process that I've done many, many hours. But basically what we do is take the brains of chickadees after they have died, and we'll slice it up into small slices. And if those birds heard particular sounds before they died, different parts of their brain will react to the stain that we use. So we specifically label zinc, which is both a gene and a protein produced by the gene, whether it's capitalized or not, because that's what's confusing. But when you go through that process, you can look at which part of the brain is active and how it changes over time. So if we're looking at the brain of a bird who heard the sound really recently, then it's going to be sort of lower towards the center of the brain, whereas if they heard it longer past, it would be moving outward towards the outer surface. And so you can see based on how that changes over time, what pathways cells are active and expressing that particular protein to see what activity is happening. So autoradiography is the same basic idea, and there's tons of different ways you can do this. You can have different proteins that you target, or different byproducts, or whatever. As long as you can tell a particular stain what to look for, you can then look for where that stain has bound. So that's hopefully a little bit more interesting than what the textbook goes into. We can also use immunocytochemistry, where you use antibodies that are attached to dye to cell components. If you've done a cell bio course, you may have made these in a lab, and they'll have like rabbit anti-goat or whatever they've labeled their antibodies as. on where it was made and which animal they've come from. We have antibodies in our body, so it's a naturally occurring process, and we can sort of hijack that system to label specific things. So we can have antibodies that look for a very particular receptor and bind that receptor, and if those antibodies happen to be attached to a fluorescent tag, then when our cells are exposed to fluorescence, they're going to light up if they have that particular receptor. So there's lots of neat little ways that we can get into this. And as I said, I'm keeping it really, really high level because we could spend entire classes on this, but we shouldn't. Um, so more things that we can do within the cell on that sort of smaller level. We talked very briefly in the text about in situ hybridization. And that's when you have the construction of DNA strands that complement an mRNA. So let me make sure I say this right. Okay, so that's when you have sort of one, let me take a step back from the slide. When you're making, or when you have DNA, when things are going together like that, you have your double strands. They are opposite each other and they sort of nest together because they link together. In order to produce proteins, that DNA separates and it's going to form mRNA, so messenger RNA. And that's because our DNA, sorry, I thought I put this on silent. Okay, sorry about that, guys. So our DNA is in the nucleus of our cells and it can't leave. And that's a good thing. It protects the DNA when it's stuck in the nucleus. But in order to produce proteins, that information needs to be outside of the nucleus where all of the mechanisms are together. So that's where mRNA comes in, the messenger. So it is going to move from inside the nucleus outside the nucleus. So this hybridization is when that messenger mRNA will extend along the DNA. It's going to be built up using mechanisms we don't really care that much about in order to assemble a complementary strand that goes along with our DNA. So the DNA acts as a template that tells what order the mRNA should be assembled into. And then once it's assembled, it can be detached and sent outside of the nucleus to be turned into protein. And so we can start sort of making use of some of this stuff in different techniques. So maybe you wanted to insert a new gene, or maybe you wanted to see if a particular mRNA strand is being expressed somewhere. So my example of zinc, the protein, you can look for the particular protein, or we could just look for the mRNA that codes for zinc proteins. So if we could flag that, if we could hybridize with just that mRNA, and that could potentially be labeled in some way, we could see things that way as well. And another way we can do things like this is if we can use that mRNA, we can tell when it's there, we can kind of use the template itself and make other copies to see what's present in a cell. That's a little bit more advanced, but it's kind of like very small scale DNA sequencing. So there's lots of ways you can do that to figure out what is currently active in the cell. But that's just because I enjoy those topics, not necessarily because they're super important here. Okay, so that's enough DNA and inside the cell stuff. Let's talk about the actual aging process. How do we look at these things? If we're staining things, we're going to need to look at what we've stained. And I mentioned early on that there were some limitations, especially with technology, where something like light microscopy, so just a normal lens and light microscope, even the best one that you can buy nowadays, you can only get about 1,500 times magnification. And that's phenomenal. That's really, really good. It wasn't always this good. We didn't necessarily have the technology to grind accurate lenses. We didn't have perfect mirrors, and we didn't have specifically refined light sources. But nowadays, there's some general consensus that we seem to have reached our limit, light microscopy. So it's just, that's as good as it's going to get based on how we understand how light and lenses work. However, if we go into something like electron microscopy, where we start using electrons and sending beams of electrons through these substances, we get a massive jump in resolution. So we can have a 2,000 or no 250,000 times magnification with a electron microscope, which is massive, absolutely massive. So we can have different kinds, transmission electron microscope and scanning electron microscope. When I was an undergraduate, we had one. It was in the basement of a university that was built in the 1800s, so you can imagine how dingy that was. And no one was allowed to touch it except for one grad student who I don't think had ever left that building, and he knew exactly how you had to work it. But in order to do this, you would basically take the cells of interest, and you only needed a couple of cells because it's magnifying so dramatically, you don't need a huge sample. And what they would do is they would embed it in a kind of resin and take very thin slices and mount those slices on the end of a tiny little rod that would then be placed in the microscope room. And it had a whole room because it needed it. And it would take very fine images of whatever you had placed there. And it was such an art that he ran everyone's sample for the department. I haven't it's a little bit less fiddly and possibly more accurate ritual and hope that the weather is good. But that's where my experience of it ended. And so the basic process of how this works, again, it's really complex and we could go into that. But we focus on the fact that different parts of the tissue are going to either block or absorb, somehow differentially interact with the electrons that are passing through. So some structures will absorb electrons and they'll appear brighter. Some will let them pass through and appear darker. Some will be somewhere in the middle. And so that allows us to get images kind of like this, where you can see different structures and that differential absorption, giving us a really nice view of the outside of a cell. So it says here, oh, so the white stuff, the stuff on the outside here, these are all glial cells. So those are our support structures and our sort of function enhancers, which is really cool. We can also have a scanning electron microscope, so still using electrons that are passing through the specimen of interest. And this is sort of an enhancement of the transmission electron microscope technique. So the earlier one would be the older version. A scanning electron microscope is going to be much more accurate. And they're saying here that this new technology can magnify images 2 to 3 million times, which is massive. I don't care too much about the numbers, but I just thought it was neat to include. And here, instead of just getting sort of a visual representation of 2 dimensions, you can now get 3-dimensional images. So this has a sort of scanning laser microscope and 2 photon microscopes. You have 2 things looking at it at once in order to get that extra dimensionality. So very neat technique. We won't do tons of looking at microscopy, but it is nice to know that these exist. What we will care a lot more about are the ways that we can measure and manipulate brain activity. So a lot of the basic techniques for brain imaging, these are the ones that we're going to see a lot of and talk a lot about. So these will go into a little bit more detail. But again, if you take an intro class, which you have to have been here, this will be all review. So we have an electroencephalogram, EEG. And what I typically do is when I'm naming things like this on a test is I'll include both the long version and the short version, so it doesn't matter which you remember. Some of them are a little wordy. But this is where we have sort of electronic, we have amplifying and detecting electrical activity of all of the neurons between 2 electrodes. So we have electrodes that are placed on the scalp, or if you have intracranial, you have inside the scalp, underneath the skin, and possible on the surface of the brain itself for maximum resolution. But these electrodes are going to pick up the tiny electrical signals of neural impulses, and they're going to amplify them so that we can see them outside of the brain. So if you've seen sort of an image like this, we have electrodes on the scalp. Nowadays, they're usually attached to a cap so that you don't have to place each individual electrode independently. If you need precision, then individual placement is preferred, but an electrode cap is just fine. And they'll settle it on your head, usually using a gel so that you get good connectivity between the electrodes and your scalp, and then they'll pick it up that way. Yep. Not nowadays. They used to be, yes. Nowadays, with the gel for contacts, they can get a decent enough amplification. And it basically depends on how important it is for your work to be accurate. So if you're doing like you need high precision, then maybe it's okay to have the gel and individually place electrodes exactly where they should be. You wouldn't want a cap, but you might have to get some participants who are willing to shave their head. For most studies where we're just looking for general area of activity, they don't care that much, and a cap in the approximately right spot is going to be fine. But it's all about what you're studying and how much precision you require. And then for me, each electrode is in a particular place. You know exactly where they are relative to the others, and each one is going to have an output. So here we can see sort of the ups and downs showing differences in electrical activity in different parts of the brain. So we can have high voltage or low voltage. We can have high frequency where they're very close together, or low frequency where they're spread apart. When we're looking at a graph like this, we're looking at time on that x-axis, and we're looking at voltage or charge on the y-axis. So that's the vertical one. So larger, higher peaks means a larger voltage, and more spread out peaks means that you have a lower frequency, which means fewer peaks per second or per unit time. And so we can interpret this combination of sort of high activity and low activity and high amplitude and low amplitude and all of those things because we know exactly where each of these electrodes are placed, and we can actually turn it into an overall map of where we're seeing higher levels of activity over time. And I don't think the textbook talks about this, but again, I TA'd for Kyle Mathewson for a couple of semesters, so I know far more about this than I want to. But when you're doing this kind of research, you very rarely rely on a single trial. In fact, you very rarely rely on even a big trial. You want 200, 300, maybe 1,000 trials so that you can average all of that data together. And that's because within our brains, we see a lot of that activity, we see a lot of variation and variability. So you want to get as many points of data that you can average across as possible. And that's how they'll get these color maps. So red would be high activity, blue would be low activity, and then sort of yellowish green is somewhere in the middle. So they would say that there's lots and lots of activity sort of in these areas when they're doing a particular language or something like that. The specifics of that study don't matter. I just wanted to show you what kind of an output you might see. And that first part labeled as A that shows you that graph, that would actually be one of these here, an event-related potential. So when you've averaged many of those recorded responses to a stimulus, you can create this nice smooth curve. So maybe they're looking at how you react to hearing a particular sound. And this would be looking at just a single electrode. And you could do this for each and every one of the electrodes to see if there's a difference. In a paper, they're usually going to report the 1 or 2 electrodes that perform best, especially for things like sound or, yeah, mostly sound. You'd get them bilaterally, so one on the left and one on the right. But this is averaging everything together. So you hear the sound, there's a moment for us to register it, and then they have an increase in activity and then a decrease in activity. And it shows us how long it takes for that reaction to occur. So we can tell somebody's reaction time to different kinds of stimuli based on when we see these sort of peaks and valleys after the event has occurred. So that's one of those really important things that we'll look at. So those are event related. And as I said, we're getting many different trials that are averaged together in order to try and cancel out that background noise, the extra stuff that goes on in our very active brains. And this helps show off the brain's unique response to different stimuli. So some people have faster reaction times, some people have slower reaction times. And if we look at them across multiple electrodes, we can figure out which areas receive that signal first, which is very cool. And if we want to talk about some of the benefits here, the temporal resolution of the EEG is phenomenal. So we can get events as close as 1 millisecond apart being so we're getting immediate reactions because it's directly tapping into those changes in voltage at the neuron level. So it's a very fast, direct way of measuring those things, because most of our other techniques are fairly slow. Spatial resolution is another one of those things where it depends. So if you're using a more precise method, so maybe you're placing individual electrodes, you're going to have better spatial resolution. The best spatial resolution, as I said, is if you place those electrodes directly on the surface of the brain. So say someone was doing brain surgery and they wanted to make sure that while they were working, they weren't damaging the language center of the brain. So maybe they put a mesh on that part of the brain to manage or monitor output while they're working or something like that. So you can have direct application for highest spatial resolution, but we can go fairly imprecise depending on how much time you want to spend positioning individual electrodes. So that'll just depend on what kind of research you're doing. All right. So another thing that they'll use that's actually fairly similar to EEGs is placing individual probes within the brain. So this is mostly done in non-human animals. They'll use a stereotaxic instrument, which is a device that allows precise positioning of a probe within the brain. The textbook has a couple of images of this, but it's basically a sort of braced stand that allows you to control the angle of a probe in all directions so that you can apply it at the exact depth and location that you want it to. This is mostly used in older research. I haven't seen as much of this lately. This would be in, say, a rat lab where they work with tons and tons of rats and they want to measure things on, say, the neuron level. So they're going to use that stereotaxic instrument to precisely line up with the thalamus of the brain. And then they're going to place probes that way or something like that. And the probes are usually very fine wire electrodes, so these can be minuscule. And again, as technology is advanced, we can now make them so small that they can be inserted into individual neurons, which means that we can have single neuron recording telling us when this specific cell is firing. And if they're inserted in the right way, you don't necessarily get information about the ones around it. Whereas if we just have sort of general electrical probes, we're going to get activity in all of the surrounding area as well. So we have, again, varying levels of precision here. And these don't just monitor. If you've set it up properly, you can also send an electrical current and cause stimulation. So you can trick the neuron into thinking that it's having an action potential, even if it isn't, and it will trigger other things to have action potentials too. So by artificially inducing that change in charge, you can trigger an action potential and see what happens as it cascades. So it's a very powerful technique. I'm actually just having a moment. What time does class end? 10. Oh, okay, then we're doing really good. It's just one of those days where I'm not keeping track. Okay, perfect. Yeah, I'll keep going just a little bit, at least until we hit the halfway. Okay, so that's the electrical stimulation and monitoring. Really, really neat. We can also have optogenic techniques. And this is one that's really cool, and I've only seen it talked about fairly recently. It's only really made it into the textbooks in the last 5 years or so because it's only just now becoming widely used. We don't go into it in detail again, but they basically have light sensitive channels that get inserted into particular. So we have, there's a bunch of single celled organisms, usually algae or bacteria, that naturally relax on light. So if they're exposed to light, they have these protein channels that open up and allow certain things to pass through into the cell. So kind of like our sodium ion channels that would open up and trigger an action potential. We can have those now light activated. So if we do some genetic voodoo in the background, we can set it up so that when we shine a light on a particular part of the brain, we can turn on those channels whether the cell gets directly triggered or not. And so here, if we've set it up properly, only neurons in particular areas will actually uptake these proteins. So they'll be the only ones that have that particular channel. And that way we can shine the light on the whole thing if we needed to, and only the areas that have these activated by light cells would then be triggered. So we have really fine, precise control of what parts of the brain are activated or led to have action potentials based on how we set this up in the background. So with our electrical stimulation, unless you're really, really careful or have a lot of time to place individual probes and you're very careful that there's no contact between cells, you're not going to get this level of precision. So this is a more reliable way to be precise all the time. And there's lots of really neat ways that they've used this already. It's becoming more and more popular, but it's helping us understand pathways in things like management and stuff like that. So that'll be one of those fields that gets bigger and bigger as time goes on. We can also have something, again, a little bit less precise, going with chemical stimulation. So you can use cannulation, basically inserting a tube through the skull to the brain and allowing chemicals to be applied directly to the brain. If we think of those chemicals as things like neurotransmitters, then we could see how neurotransmitters in parts of the brain change something about their behavior. You can also have microdialysis, which is a more complex but smaller version of cannulation. So instead of just having a tube that sits through the skull and onto the surface of the brain, you can target particular areas with a lot more controlled dosages. But again, it's basically seeing how do different parts of the brain activate or react in response to different neurotransmitters and other chemicals that might mimic neurotransmitters. So we could see for some of those neurotransmitters, the pesky ones that are sometimes excitatory and sometimes inhibitory, you could use them in different areas and see if we have an excitatory or inhibitory response to figure out what parts respond which way. So lots of possible methods to make use of that. Continuing on into sort of other ways that we can study the brain, mostly now starting to get into how areas of the brain on a larger scale react or interact with function and behavior. We can look at things like natural experiments. So this is a little bit strange because they're talking about how patients who had brain damage were used to determine which parts of the brain were related to which functions. But it's labeled as an experiment, even though they were looking at people who just happened to be brain damaged. Um, there were, of course, other experiments that were done where people received damage intentionally that probably fall more accurately under an experimental context. But a lot of what we originally knew about how the brain works came from case studies on individuals who had damage from brain trauma of some kind, and then we would look at how their behavior had changed after that damage. So if you've heard of Phineas Gage and the piece of steel through his frontal lobe, that was a massive change in his personality afterwards. And so that gave us the indication that that part of the brain is probably linked to personality. There's all sorts of different case studies that give us that information. Again, we need to keep some of those limitations in mind. So the damage might happen to affect areas that cause changes in behavior. It might impede function or it might not. There's also sort of the variability between individuals. We've talked a little bit about neuroplasticity and the fact that we can recover after damage. So if they're only looking at people's brains after they've died, maybe years later, then it might not be an accurate representation of ability versus damage if they've had some recovery since. Again, if you're not physically manipulating the situation, it's very hard to make those causal conclusions. But we can look at sort of what are some of the possibilities, what are some of the associations. And it's also interesting to note that sometimes you have full damage to an area, or maybe you'll have complete damage to an area. Again, if we're not controlling this damage, we're just relying on accidental damage, then we might see complete damage to Broca's area, or maybe partial damage to Broca's area. But that can give us useful information to see how does functionality change between partial and complete obstruction. For more experiment equivalent type responses, for things that work with the word, we could look at ablation or lesioning. So ablation is a surgical technique that involves the removal of brain tissue. This is very rarely used in humans. There are situations where it has to happen, say you're removing a tumor or a part of the brain that's functioning improperly and causing harm. Maybe we can look at things like removing part of the corpus callosum or something like that. So it can happen, but it very rarely occurs, especially with an attempt for an experimental study. So with ablation, it's typically used on fairly large areas. So it's pretty imprecise unless you have sort of a very accurate way of determining where the hard edge should be. And again, with our neurons being so intertwined, it's very hard to remove just one specific area without affecting the surrounding area. And most ablation, especially in research, is often done by aspiration, which basically means you apply a tube and suction, which is a pretty traumatic way of doing it. You can also have lesioning, where you surgically damage the neural tissue. So this is usually precise damage. This would be an or pulling something out entirely. This would be causing damage at this particular spot, probably using a probe and either I think I've seen it done with heat or cold, or even just slicing in a particular place. And some of that damage can be reversible. So if you only sever, say, a single axon in a place where axons could theoretically regrow, you could then watch that repair happen. And I might have mentioned it before, but I know someone who, for her master's research, was looking at a way of applying a cold signal to the brain, so freezing a localized area by something inserted in the scalp that would deactivate all of those cells in that area because they were too cold to function. But when the cold was turned off and the area was allowed to reach normal temperature, the function would return. So as long as you deactivated that part of the brain for very short periods, you could have temporary and entirely reversible lesioning, which was really neat. Where she was looking at, I think it was song production in songbirds. You could sort of turn on and off part of the brain that was responsible for their ability to sing and look at the effects and then bring that behavior back, which was really, really interesting. So again, lots of upcoming research in this area. All right, so the next of our series of imaging techniques are when we're looking at brain imaging. But before we do that, transcranial magnetic stimulation, EMS. And this always gets grouped in with the brain imaging techniques, but it isn't necessarily an imaging technique. This falls under more manipulating brain activity. So uh, from the name alone, you can kind of get a feel for what's going on here. So transcranial, meaning across the cranium, across the skull. So we are using something from outside the brain to affect something inside the brain. And we're using magnetic stimulation across. So we can have a non-invasive technique where we expose the brain to a particular magnet coil in order to induce voltage on the inside of the skull. So our neurons fire through changes in electrical activity. As they're firing, they're creating that change, and as there's a difference in potential, we can trigger action potentials. So you can actually use this coil to induce changes in voltage that can either cause firing or, if you do it right, disable firing in particular areas. So sometimes here, when they're having increased firing, it's not firing in a particular pattern. So if we don't have pattern and order, the brain can't make sense of what's happening, so we don't have the functionality of that part of the brain. So I've had students who have trouble figuring out how increased activity reduces functionality, and it's because it's increased activity pattern with no usable function to it. And so you can hold this device over different parts of the scalp. You can have it turned on and off at differing rates and different patterns, and look at how people's behavior changes when different parts of the brain are on. So a lot of these studies will look at things like how quickly do you respond to an image on the screen? Maybe they're showing you shape, and anything that's a circle, you have to push the left button, and anything that's a star, you have to push the right button. Well, what happens if they interfere with the back of your skull, with that occipital area, where we're processing information? Does that slow down your ability to decide if it's a star or a circle? What's going on? And so if they can precisely disable different pieces, we can figure out where that function is tied to specific. All right. Oh, and this is an interesting one. It's not just useful for research. There's also some evidence that it can be useful therapeutically. So using this transcranial magnetic stimulation, things like, I believe lately it's been used to treat anxiety, and I believe I've seen it as helping people process emotions better. But it's, again, a fairly recent technique. Yep. It might, but it's not a long-term change because it's just a brief exposure, and it's not like it's not directly touching anything. It's not sending a signal by physical contact, so it's a lot less intensive. Yeah, so similar idea, but less extreme. All right. And so for the last couple of techniques, we're going to look very briefly at computed tomography. I've seen this called lots of different things. Computed tomography, computerized tomography. You'll see it shortened to CT or CAT. So it can be a CT scan or a CAT scan. All the same thing, just with different labels. And this is where you're producing a series of X-rays. You take a whole bunch of slices and then you put them together. You composite them together into a three-dimensional image. You're usually going to add false colors for contrast. So they will usually light up areas in a particular color to show something of importance. But mostly we're relying on the fact that we get differential absorption of those X-rays in different parts of the body. So we can see different densities within all of the different blood vessels in our brain. We can see where is high density tissue versus low density tissue, and so on. So look at the next slide. There's a couple of images here of what you've probably seen many, many times in medical dramas and stuff like that, sitting in a CT machine, and then also the scans that it can produce. Similar, but not quite the same, we can have MRIs or magnetic resonance imaging, as well as a couple of the subcategories of MRI. And so this is a way of measuring radio frequency waves, excuse me, that are emitted by hydrogen atoms that have been exposed to a magnetic field. So if you've ever watched any medical drama, when they have an MRI machine, there's always an episode where someone brings metal into the room and it gets trapped in that magnetic field and they have to go into the machine and turn it off. That's what's going on in one of those rooms. So it looks similar to a CT, but it's all set up through magnets. Oh yeah, it's a big thing. And even something as simple as like my mom has two artificial hips, so you have to be very aware of that. And even like lots of metals in the blood, there's certain things you got to watch for. So there's usually an extensive screening procedure before they do that. But yeah, there's lots of horror stories of stuff gone wrong. Um, with the imaging here, we're going to see a lot of response specifically because of hydrogen atoms that are in water molecules. So MRI is going to find a lot to do with the water that's in our brain. And that kind of makes sense. Our blood is mostly water, so it's going to light up and show us where blood is within our brain. But they've gotten a lot better recently. They're getting more accurate, a lot more detailed, and now they don't just see hydrogen emissions at the magnetic field. Now they can start looking at other elements as well, which will give us even more detail and even more things that we can look for. For our subvariants of MRI, we can have DTI, which is diffuser tensor imaging, which is basically where you're using an MRI, but you're focusing on the movement of those water particles. So if I hop to the next slide here, the one that's labeled B that shows all those different tracts, that's showing movement of blood along with some of the pathways in the brain. So that's what DTI looks like. Whereas the first one, A, would be an MRI, and it's highlighting a tumor at the top there. We can also look at PET scans, positron emission tomography. This is how we can observe brain regions through a radioactive substance that's injected into the bloodstream. Um, People always get nervous when they see that it's radioactive, but this is something that's usually lowly radioactive. It's going to have go away quickly and isn't going to cause lots of problems. So they've picked a safe, mildly radioactive substance, which is usually glossed over in these descriptors, but bring it into the bloodstream, and then we get to watch where those substances are moving around in the brain. So you look at where those radioactive particles are congregating in the brain. So the scanner is going to pick up the emissions from those radioactive substances, and they're going to form a color-coded image based on density. So it's going to indicate relative activity of brain regions. So if a part of your brain is more active, it's going to be taking in more blood because it needs more resources. And if it's taking in more blood, then it's going to have more of those radioactive isotopes in that area. So the problem with PET scans is that you have to have these changes last at least 30 seconds or more. So very poor time resolution here. Um, so we only get sort of prolonged changes, areas that are active for long periods of time. And because this is only looking at where things have concentrated, this doesn't actually give you an image of the whole brain. It just gives us areas of activity. So you'd usually have to do an MRI and then overlay PET scan results on top to figure out what structures that corresponds to. So it doesn't actually give you an image of the brain, just the parts of the brain that are active. So a lot of these have and this is what one of those scans might look like imposed on an image of the brain. And then I think this is the last one. Yeah, fMRI or functional magnetic resonance imaging. And this is again, sort of sub part of our MRIs, but it's looking at increases in blood flow and oxygen usage within the brain. So again, MRIs are looking at sort of those hydrogen molecules, but also now looking for oxygen usage as well. So where is active in your brain, where is pulling blood to it, and where is using up the most resource. So this is something that's really good for repeated measurement. So it doesn't have the short lifespan that you might have with a PET scan. It's a good alternative to PET scans because it gives you good spatial resolution and it's a little bit less laggy in terms of response time. But it's really expensive because you need a highly specialized MRI machine. So there's sort of trade-offs on it's a better technique, but it's also more costly. So we still see PET scans in areas where they can tolerate that lag. And then this is just an example of what an fMRI looks like. And another thing that our textbook does phenomenally well is that it has these nice little summary charts after these long stretches of multiple techniques. So it talks about the difference between EEG and CT and MRI scans and fMRI. And it talks about time, spatial resolution with a nice convenient description. So diagrams like this are usually useful to keep around, which is why I highlighted it in the text here. And then I have a conclusion slide for brain imaging techniques. And so, as I said, when we started talking about brain imaging, we care a lot more about brain imaging than we did about, say, the genetic manipulation side of things. And that's because there are other classes that do that. So we kind of hand wave a little bit of the genetics side of thing, but we do really care a lot about brain imaging because it's one of the main experimental techniques used in neuroscience. And especially as the technology is coming along, we get to see more and more numbers of studies, more accurate studies, and studies that have a lot more flexibility to them. But we do need to keep in mind the limitations. So if we talk about, we mentioned that our PET scans have a very bad time resolution. So that's something to keep in mind. fMRI isn't necessarily as sensitive, especially in structural things, because it's looking at changes in blood flow and activity. Um, and so we want to look at stuff that way. We also kind of hinted at the fact that retest reliability is often low. When we were talking about EEGs, and I mentioned how you have to do hundreds, if not thousands, of trials to get a nice smooth image of noise, a lot of these techniques, um, especially the ones that are a lot more expensive than EEG, you aren't necessarily capable of running tests to average your results. And so if you only run a test twice, you might get very, very different results and not be sure which one is accurate. So we need to keep that in mind. And then for the researchers on the other side of the scale, there's some issues with methods of data selection. The textbook goes a lot more into detail about this, but there's a lot of controversy around sort of picking and choosing the best piece of data or the best scan to use for a particular study. So if you're doing something like EEG, where you have 100 electrodes, well, you're only going to really report the best one, the one that shows the best, even if it's not necessarily an accurate representation of the activity overall. So there's some issues there when reading stuff about how they've chosen what to display and what to talk about. So just some critical thinking there. And that is a perfect spot to stop. We only have maybe 15 or 16 slides left, so we can finish that up on Thursday. And as I said, on Thursday, we're also going to go over the written assignment. I'll make sure I have my slides together to chat about that. If you're curious and want to look at it in advance, you can absolutely do that. All the details are up online and it's not complicated. But yeah, we'll talk on Thursday.